The overall goal of this video is to present an electrokinetic technique called isotachophoresis, abbreviated ITP, and its microfluidic applications for sensitive detection and separation of ions. First, the physical principles underlying ITP are discussed, in addition to the two standard modes of operation, namely peak mode and plateau mode ITP. On-chip ITP is then demonstrated with 3 experiments, beginning with a simple protocol for sample preconcentration, where fluorophores are detected by focusing them in a sharp peak. Second, an ITP assay used for on-chip sample preparation is shown where DNA and RNA are selectively extracted from bacterial cell lysate. Finally, a label-free detection assay is used to purify and quantitate amino acids using plateau mode ITP. Ultimately, amino acid purification, analyte preconcentration, and selective extraction of nucleic acids can be achieved through ITP.
Today we will introduce you to the underlying physics and methodology of a versatile electrokinetic technique called isotachophoresis.
Our group has pioneered ITP methods for label-free detection, sample preparation, and rapid hybridization assays.
We will share with you 3 representative assays that demonstrate the ability of ITP to preconcentrate analytes, extract nucleic acids, and purify and detect amino acids.
These assays highlight the versatility of ITP and demonstrate the simple steps needed to ensure repeatable experiments in the lab.
In ITP, sample ions selectively focus at the interface between a leading and trailing electrolyte, abbreviated LE and TE respectively. The sample ions travel through the microchannel at a constant speed determined by the velocity of the leading ions. Sample ions focus if their effective electrophoretic mobility is bracketed by the mobility.